In this audio lecture, we're going to be talking about acid-base balance, and really this is an introduction for the acid-base disorders in the follow-up lectures. Acid-base balance is an important aspect that we need to maintain control of, because really small deviations in pH, which is what we measure acid bases in, can create tremendous disruptions. And so we'd like to maintain a pH between 7.35 and 7.45. Okay. And so we have multiple aspects of this that we need to control for. So we can see that we maintain pH through multiple systems. We use body fluids. The kidneys are heavily involved in pH regulation, and also the lungs participate in this pH maintenance as well. And so if we look at this figure over here, you can see that really hydrogen, which is what pH is actually measuring. So pH is a measurement of hydrogen concentration, and as the hydrogen ions go up, pH decreases. As hydrogen ions go down in concentration, pH increases. Right? So we can see that we're a little bit alkaline in our normal pH already, but really we like to maintain pH in this narrow range. So we can see that hydrogen ions and bicarbonate ions are involved in this. We can see that hydrogen ions are a product of metabolism, right? So they're involved. They are produced in the consumption or production of ATP. Okay? So is carbon dioxide. We can see other acids here are also produced in this process, and we'll talk about the different types of acids in a second. We have three primary mechanisms in buffering changes in this pH, and we could use chemicals, we could use the lungs like we said before, or we could use the kidneys, and we'll talk more about each system in more detail coming up. Okay. So we can see that acid-base balance, some causes of it are going to be organ failure. Okay, so respiratory, if your lungs stop working as efficiently, it doesn't necessarily need to be organ failure, because you die if you can't breathe, you die if your heart can't beat, and then if your kidneys can't filter anything, like we see in chronic kidney disease, we can see that eventually leads to pH imbalances. But even short-term disorders like emphysema isn't necessarily something that's going to kill you right away. It's going to take years to really create a detrimental effect. And we can see that malfunction of these organ systems also creates these pH changes. Okay. Infection, organ trauma, which can cause disruption in normal function, can all be sources of these pH imbalances. So generally we consider two different types of acids. Okay, so we consider volatile and non-volatile acids, right? So volatile acids are something that can change form, so it changes. So here we have, and this H2CO3 is called carbonic acid, and so as this carbonic acid, so and it's a part of a bigger equation. So the carbonic acid bicarbonate buffer system. So this carbonic acid can actually turn into a gas, and this gas is carbon dioxide. And then the gas can be released from the lungs, and that's considered a volatile acid. Now, non-volatile acids cannot be excreted in the lungs, and so we see these gases take the form of hydrochloric acid, sulfuric acid, or phosphoric acid right here, right? And so we use these acids, these non-volatile acids. We secrete non-volatile acids because they can be converted into a gas. We secrete them in the kidneys, into the urine. Okay, so carbon dioxide is naturally a product of aerobic metabolism, right? So it's a byproduct of ATP synthesis. It's produced in the Krebs cycle, okay? The second stage of ATP production. So we see some chemical buffers. So this is more. These chemical buffers are going to prevent changes in pH, and specifically we have the bicarbonate buffer system, which is also called the bicarbonate carbonic acid buffer system, and that equation is carbon dioxide plus water. It's converted into carbonic acid through the help of carbonic anhydrase. Carbonic acid, when it forms, quickly disassociates into hydrogen and bicarbonate. And so we see that our body fluids have this carbonic anhydrase to facilitate the conversion of carbon dioxide into something that we can more efficiently carry. So we have it in the extracellular fluid, we have it in the kidneys, we have it in the lungs, we also have it in the hemoglobin, right? And the hemoglobin, its job is not only to transport oxygen, but also to transport carbon dioxide in the form of hydrogen. Phosphate buffer systems. So the phosphate buffer systems binds free hydrogens to phosphates. So phosphates are an anion. They are high in concentration in the intracellular fluid, and so hydrogen production is sequestered by binding these phosphates. So we can prevent the effects of an increase in hydrogen production by binding to the phosphate group. Now, proteins are really a predominant, this is the most abundant resource. The bicarbonate carbonic acid buffer system is really the most utilized. So the proteins are abundant inside of the cells. We have less proteins outside of the cell. The main protein outside of the cell is albumin, and then inside of the cell we have a number of proteins, right? So we can use these proteins to take up and bind extra hydrogen ions that are free that are floating free, right? And by binding them to proteins, we can actually prevent the decrease in pH that we would see with a free hydrogen. Now, in the same note, the proteins can also be used as a hydrogen reservoir, and so they actually are proton reservoirs. So hydrogens are also called protons because they are missing a negative charge. They are missing an electron, and they only have one electron to begin with, so we can call them protons as another term. And so these proteins can actually release hydrogen ions if the pH begins to increase too much. We also have hydrogen potassium antiporters that move hydrogens between the extracellular fluid and the intracellular fluid, or even the transcellular fluid, so like the serous membranes. So hydrogen and potassium are exchanged. That is what an antiporter is, and to facilitate that movement, we need to use ATP in that process. Okay. These can buffer changes in pH momentarily, and these are going to be limited in how they can be used. If these buffers can't buffer, if they can't bind that excess hydrogen, now we start to see pH changes. So here's the carbonic acid bicarbonate buffer system. Again, this is the primary extracellular fluid buffer system. It is also found in hemoglobin. And it's important in the kidneys and lungs as well, because in the lungs we are excreting carbon dioxide, and then in the kidneys we're excreting really hydrogen, right? So we are really concerned about formation of carbon dioxide and hydrogen to be able to easily secrete those two substances. So in the lungs, we're going to move this equation to the left, so it's a reversible equation. So really, if we get an increase in carbon dioxide, now it's going to balance out, and this is called stoichiometry. So stoichiometry is really, this equation likes to be in balance, and so if we get an increase in carbon dioxide, we see an increase in carbonic acid, and then we'll see an increased presence of hydrogen as well. And so you might be saying, well, bicarbonate's produced, so what effect would this hydrogen have? Well, remember that CO2 is not our only source of hydrogen, and so we also produce hydrogen in the form of lactic acid, of keto acids, and so we have additional acids being put into the system. And so if you start to see an increase in carbon dioxide, now this is going to lead to an increase in hydrogen. And remember that bicarbonate is going to bind some additional hydrogen as well, and so the increase in hydrogen can increase or decrease the pH. Right, and then in the kidneys, we're going to favor really the formation over here. So if this carbon dioxide is increasing in circulation, now we want to shift this equation to the right, and by secreting hydrogen in the kidneys, we can really we can continue favoring this equation to move to the right here. Right? Whereas in the lungs, where we're expelling carbon dioxide, we're moving this equation to the left. So in the kidneys, you're moving it to the right, and then in the lungs, you're moving it to the left, and then in the red blood cells that are important in carrying this carbon dioxide. So remember that carbon dioxide is carried in the form of hydrogen bound to hemoglobin in the blood, and the hemoglobin specifically. So we form this hydrogen, and then we actually move through a chloride shift, right? So remember the chloride shift, that bicarbonate is moved out of the red blood cell, and then chloride is moved into it to keep electrically neutral. In all these places, we have really this carbonic anhydrase, this enzyme that facilitates this reaction. So carbonic anhydrase is important in this conversion right here. So this conversion of carbon dioxide to carbonic acid, right? So carbonic acid naturally disassociates it. It's called an acid because it donates a hydrogen group here. We have this carbonic acid in the lungs. This allows for carbon dioxide production, and then in the kidneys, we get the reverse reaction, and that we want to favor really the formation of hydrogen. This allows us to excrete the hydrogen in the lungs, or in the kidneys. Right. And through that, you are decreasing the level of carbon dioxide. Okay. So this is carbonic acid is considered a weak acid, and then bicarbonate is considered a weak base. So bicarbonate is considered a base because it binds a hydrogen group, and when it binds a hydrogen, it turns into carbonic acid. So weak bases always turn into weak acids, and weak acids turn into weak bases when they donate that hydrogen group. All right, so a little bit more about the hydrogen potassium antiporter. Here we have a cell. Now remember that in our cells we have a high concentration of potassium. Okay? And when we get an increase in hydrogen ions or protons in the extracellular fluid, we want to buffer these hydrogen ions by moving them into the cell, right? And so we can do that by exchanging it for potassium. So in this hydrogen potassium antiporter, so we're moving them in two different directions. We are moving potassium out of the cell and exchanging it for hydrogen, right? So this is limited capacity because as we move that hydrogen inside of the cell, now the pH is decreasing in the cell, and now the proteins that are in that cell are also going to probably bind that hydrogen. But as soon as the proteins are really saturated, now the pH is going to start to decrease, and then the proteins would start to unfold, and they would start to lose function. On the other hand, if we see that there's this excess potassium in the extracellular fluid, which is a condition called hyperkalemia, we want to move hydrogen out of the cell and then move potassium into the cell. Now this process right here requires ATP to be used because moving potassium against its concentration gradient is something that requires energy, and ATP is that energy in our body. So here we have protein buffers, and so the protein buffers, remember proteins are made up of amino acids, and so these amino acids can accept or release hydrogen ions. By binding hydrogen ions, we are preventing a decrease in pH, and by releasing a hydrogen, which they would do if we are in if pH is starting to increase, okay, now these amino acids can release this hydrogen group. Now remember, these are found primarily intracellularly. We have them extracellularly, but they are lower in concentration extracellular compared to intracellularly. So carbon dioxide diffuses across cell membranes, and hydrogen requires a channel because it is a charged ion. So it does so through a channel, and that's called facilitated diffusion. But carbon dioxide freely diffuses into and out of cells, but inside of the cells, carbon dioxide is broken down into bicarbonate and hydrogen ions through the conversion of carbon dioxide to carbonic acid. Okay. Now remember that we do have proteins in circulation. The primary one is going to be albumin, and then we have plasma globulins, which would be components of antibodies here. But really, most of this is going to occur in the intracellular fluid. So now we need to introduce this idea of compensation. Compensation is going to be how we adjust for pH disorders. And so this compensation is going to be talking about really respiratory disorders. So this is how we generally see this acid-base disturbance. We can see a respiratory origin or a metabolic origin. And so respiratory origins cannot correct themselves. They are going to be short-lived. Really, corrections, if they do correct really a pH imbalance, but we have this compensatory mechanism that needs to correct it. So respiratory disorders are corrected by the kidneys, and then metabolic disorders are corrected by both the respiratory and the kidneys, because they're not directly involved. Now, if the disorder is originating in the kidneys, now we have to really use respiratory compensation for that. Right, so that's going to, so this compensatory mechanism that corrects the pH imbalance is going to be determined by the cause of the imbalance. So if it is renal dysfunction, now our kidneys our lungs are going to have to excrete that excess hydrogen that's building up in our system, or decrease excretion. Right. So our organ systems cannot self-correct. And so here we have a table that is displaying the normal arterial blood gas values. This is going to be an important table for you guys to know. Okay, so I'm telling you that right now. Be familiar with this table and be familiar with the values that we see. So we can see that pH ranges between 7.35 to 7.45. Arterial oxygen matches the alveolar partial pressure of oxygen, which is about 100 millimeters mercury. Arterial partial pressure of carbon dioxide is really 40, but it fluctuates, so it ranges between 35 and 45 millimeters mercury. Bicarbonate, so our cells do produce so our renal cells do produce bicarbonate, and this is going to range between 22 and 26 milliequivalents. Milliequivalents is a term that's referring to a charge that corresponds to the millimolar value. But we will use milliequivalents because this is what they use in health professions. So base excess and base deficit are terms that describe how much strong acid has to be added to a solution to be able to adjust the pH back to normal. Right. So this is ranges. So I've seen it range between -2 and +2. Your textbook provides a value of -2.4 to +2.5 milliequivalents per liter. So if you become acidotic, you would need to add a strong base to that solution, and then if you become alkalotic, meaning your pH is increasing, you would need to add strong acid to that solution. So if you are alkalotic, you sit on the positive end of this, and if you are acidotic, then you need to add really a base to the solution to be able to buffer out the change. Right? And then arterial oxygen saturation is 96 to 98%. That is a typical value. So a lot of textbooks provide 100% or 99%. I've never seen it that high. It generally sits in this range, 96 to 98%. All right, so a little bit more about the compensation here. We have a change in pH. So if we get a pH that is less than, so this says 7.4, but realistically it's less than 7.35, we are considered acidotic at that point. So that is how you're going to classify this to begin with. So we want to really classify this disorder to begin with determining if it's acidosis or if it's alkalosis. And if it's a pH less than 7.35, it's going to be acidosis. And so from that, then we will determine if it is respiratory origin or metabolic in origin. And so respiratory in origin we can tell by changes in partial pressure of carbon dioxide. So this increased partial pressure of carbon dioxide would tell you that is respiratory of origin, and that's because now our lungs aren't efficiently really expelling this carbon dioxide. It's building up in the system. So that is going to be greater than 45 millimeters mercury. I apologize for these values. This image came off the internet. Over here we can see that we have a different tool that lets us determine that we have metabolic acidosis, and really this is going to be a deficit of bicarbonate. Right, so we see this decreased bicarbonate and metabolic acidosis, and we can see that. So this says that it's just respiratory origin, but that is not necessarily the case. So as we get more into each of these conditions, you can see that the compensation includes also the renal mechanisms, and so the kidneys would retain more bicarbonate and then excrete more hydrogen, whereas the lungs are going to try to excrete more carbon dioxide by altering ventilation. If we are higher than 7.45, we are considered alkalotic at that point. So, and we have the same two conditions. We either have metabolic alkalosis or respiratory alkalosis. And if we're respiratory alkalosis, this means that we are not retaining enough carbon dioxide in our blood, and we're expelling too much. And so we would expect a partial pressure of carbon dioxide to be lower than what it should be. And remember that really fluctuates between 35 and 45 millimeters mercury. So a value of about 30 millimeters mercury would tell us that we are suffering from respiratory alkalosis. Okay. And the kidneys would compensate for that by really excreting more bicarbonate to bring down the pH. Okay. For the other condition of alkalosis, so we have metabolic alkalosis, and this is going to be really an increase in bicarbonate levels. So bicarbonate, remember, is a base. Okay, so if that base is increased, now it's going to become more alkaline, and so we would want to have both renal and respiratory compensation. We can see that we slow our breathing to let the CO2 build up in our system, and so we're trying to increase the partial pressure of carbon dioxide. And then in the kidneys, we're going to really excrete less hydrogen and then reabsorb less bicarbonate. And this is going to bring the pH back down to that normal range, right? So it should be noted that the kidneys they take longer to respond to these pH changes, whereas the lungs are going to be really only effective over a 24-hour span, so they don't really efficiently control for long-term changes in mechanisms and pH disorders. And so, but the long-term mechanisms are going to be mediated by the kidneys primarily, so changes in excretion patterns. It is going to be important that we have those two systems really functioning well. So disorders in the respiratory and or the kidneys can create tremendous changes for pH imbalance. So all right, that is the end of this audio lecture. Stay tuned for each individual conditions.